Thank you very much for this introduction. So indeed, I will present you a work which is about simultaneous methylation and fusion detection for pediatric tumor characterization. My talk will be mainly based on brain tumors, but I will also have a quick word about sarcomas. So as a quick introduction, in neuro-oncology, there are many entities that can be pretty challenging to differentiate, but they are crucial to identify because they will induce completely different diagnoses, prognosis, and treatments. And within the past years, with the advance of sequencing technologies, there has been a huge improvement in brain tumor classification. And in that field, methylation has proven its undeniable impact in the help of the classification of those tumors. It has even now become a full diagnosis criteria in the WHO classification. And in that field, the gold standard of assignment for methylation are the Illumina epic array. So as I was telling you, with the latest classification of brain tumors, there is now an integrated classification that does not only rely on histological parameters, but also takes into account molecular aspects. So I put you on the list here on the slide some genes that will be particularly important to look for at the diagnosis, such as the fusion in the ZFT or YAP1 gene, or also alterations in genes such as MIB, MN1, or FOXR2, in order to have a complete characterization of the tumor. So for the brain tumors, with the methylation, we used the reference classifiers of the team of Kapper in Heidelberg, for which they studied more than 2,800 samples, and they looked at the methylation with the Illumina epic array. And once we have this classification, when we will study our tumor of interest using random forest algorithms, we will be able to determine to which tumor group it belongs. So at the lab, we developed a workflow for adaptive sampling in order to study those brain tumors. So we're going to work on frozen brain tumors, starting with 2 micrograms of genomic DNA. We prepared the libraries using the long sequencing kits 114, and we follow the protocol of Oxford Nanopore. We also implemented the fragmentations with G-tubes from Coveris with a target size of 20 kb in order to homogenize the size of the fragments in the library. So as you can see here on the flowchart, the preparation of the library is very fast and easy. Within half a day, the library is ready to be injected. Once it is prepared, we're going to divide it into 2 parts, and we're going to proceed to the injection of the first half of the library for almost 24 hours on a R10 flow cell and a MKA-1B device. Once the generation of the data reaches a stagnation, we're going to stop the run. We're going to perform a flow cell wash with a nucleus mix, and we're going to inject the second half of the library for another 24 hours. So as you know, with adaptive sampling, we can specifically target some genomic regions of interest. And in the case of brain tumors, we design the file that we call the bed file that we're going to load into the MinION software, which contains all the fusion genes or genes of interest in the brain tumor entities. And as you can see here also, we always take into account a 5 kb flanking region, upstream and downstream each gene, in order to increase the coverage of the regions. So this bed file is the one that we basically use for the study of our brain tumors. It represents 1.6% of the genome, because the recommendations are to be around 0.1 and 10%, and the closer to 0.1, the better.
Hi.
So once the sequencing data are going to be generated, we have the bioinformatic pipeline, and at the lab we have a bioinformatic team who developed a pipeline of analysis that is called NanoClid, which is published on GitHub, and you can find it on the link I put on the slide. I will not go much into details about this pipeline, but with the latest version of the software, we now use Dorado for the demultiplexing and base calling, and we use Minimap2 for the long-read alignments. For the SV calling, we use a combination of Sniffles2, Svim, QtSV, and Nanovar. And as we're interested here today for the methylation calling, this will be performed by S5C. So with adaptive sampling, as you understood, we're going to look at the fusion detection with the targeted enrichment. But with the rejected strands of DNA that will be around 400 base pair, they're going to be also very informative, because we're going to look at the methylation on the CPGs on those strands. And on average, with an adaptive sampling run, we're going to look at the methylation of 6 60,000 CPGs, which is more than enough to obtain a reliable classification. And also with those rejecting strands, we're going to obtain a shallow whole genome sequencing, which will allow us to obtain a copy number profile and to study the dynamic of the tumor. So now, since we implemented this technique at the lab, we have sequenced more than 10 frozen brain tumors, and here is the summary of the different types of brain tumors that we were able to characterize thanks to this technique. As you can see, we had 1 medulloblastoma, 3 ependymoma, 1 with a relay fusion, 2 with a YAP fusion, and some other tumor types with a MIB, MN1, CIC, or other alterations. And so now I will just present you some examples to be a bit more concrete. So the first example was a young patient who presented with a bulky right hemispheric tumor at 1 year old, and at the anatomopathology, the histologic aspects were compatible with an ependymoma. So we received the frozen brain tumor sample at the lab, and as you understood, we performed the adaptive sampling, enriching the genomic regions of interest that I showed you before. So here is the copy number profile that we obtained. And you can see there is a very good resolution, and it gives us a bit of the dynamic of the tumor. And here there was a monosomy in chromosome 22. So after this, we're going to have a look at the methylation classification. So here is the t-SNE representation with all the brain tumor groups. And on this representation, we can see where our tumor clusterizes. So if we just take a zoom at it, we can see that with the methylation data, the tumor went along the ependymoma with a YAP fusion. So with this information, we decided to take a close look at the adaptive sampling reads of the YAP fusion. And you can see here on the representation of the IGV that we were able to detect with a very good coverage the soft clip between the YAP1 and the MAMLD1 gene, which made this tumor a YAP1 fused ependymoma, and we had all the information to classify this tumor. The second example was a young patient with a frontal tumor at age 11, and the histology was in favor of a high-grade brain tumor, which was difficult to classify. So once again, we received the frozen brain tumor at the lab. We performed the adaptive sampling, which gives us the copy number profile, where we can see all the chromosomic changes of the tumor. We then take a look at the methylation, and here again, there is the t-SNE representation with all the tumor groups. And we can see here, if we take a zoom, that this tumor was a brain tumor with a CIC alteration. So knowing this, we go to see the CIC gene with the long read of adaptive samplings. And this was one of the first that we characterized, and we were able to detect the soft clip between the NUT-M1 and the CIC gene. So we once again had all the information to classify this tumor amongst the tumor CIC fused. And for the last example, I told you I would have a quick word about sarcoma. So this one was a young boy presenting at 6 years old with a tumor of the posterior fossa, for which we also received the tumor sample. We performed also the adaptive sampling with the bed file to enrich the genomic regions of interest. So here is the copy number profile that we obtained, which also had a very good resolution. And after this, we go to look at the methylation, and in that case for sarcoma is not the Kappa classifier anymore, but is the one that was developed by the team of Kirsch. But the principle remains the same, and we still use random forest algorithms. And so if we take a closer look here, we can see that this tumor was clusterizing amongst the Ewing sarcoma. And what I forgot to tell you, which was important, that at the anatomopathology, they were not able to distinguish between a medulloblastoma or an Ewing sarcoma, this kind of tumor. And so here, unlike for the brain tumors, the classification of sarcomas using nanopore sequencing has not been described yet. So in this tumor entity, there is a particularly important fusion that we want to look for, which concern the UWSO1 and the ERG gene. And so with the long read of adaptive sampling, we went to look for this tumor. And as you can see here on the IGV screenshot, that we were able to detect it with a very good coverage between both genes. So here also with the sarcoma, we had all the information needed to classify this tumor within this Ewing sarcoma group. So I convinced you that nanopore sequencing is a 3-in-1 tool for the complete characterization of brain tumors and sarcomas. as it will allow you to obtain simultaneously the copy number profile, the methylation profile, and to detect the fusion genes of interest. And all of this is performed within a very reduced turnaround time, because here, on average, it took us 10 days from the DNA extraction to the results to classify those tumors. Thank you very much for your attention, and if you have more questions after the panel discussion, please come on Friday to discuss around the poster. I will be more than happy to exchange with you. And of course, thank you to all the team in Curie who is working with me to develop this project.